Okay, welcome to the second video in the series, though you may be watching this as the first one. This one's on the Calvin cycle, as you can see. And yeah, this is the BioX video for the Calvin cycle. It's got lots of other names. It can also be called the light-independent reaction or the dark reaction. So just watch out for that, because on exam questions it could be thrown into you either way. What I'm going to do is I'm going to just show you roughly where this is happening. I'll zoom out. So we're inside a chloroplast here. So I'm just going to show you just faintly the outer membrane. Remember, it's a double membrane for a chloroplast. Just to show you, this is outside the chloroplast, but everything else is taking place inside this space here. Remember that chloroplasts also have these stacks. Each individual part of them, each individual bubble is called a thylakoid, which you can learn about in the light-dependent reactions video, and together they form the grana. But we're not looking at those now. We're going to focus in this space. It's like the cytoplasm of the chloroplast, and this area is called the stroma. So I'm just going to write this down here. I recommend that you have a similar piece of paper, or maybe a slightly smaller one that you're drawing on as I go through, so that you can track what I'm drawing. So the Calvin cycle is all about producing sugars, and those sugars can be used by the plants to make all kinds of other molecules. Let's run through it. Starting off, we have a molecule called RUBP, and I'm going to represent the fact that it has 5 carbons by drawing 5 little circles. So this is called RUBP. The full name for this molecule is ribulose biphosphate. It's useful if you're familiar with this term. But it's very unlikely that you're going to be asked ever to recall it straight from memory. But if you remember RUBP, that'll be fine. That's the standard abbreviation, a bit like ATP is for adenosine triphosphate. So what happens? RUBP is going to be reacted. At this point, carbon dioxide comes in, and carbon dioxide, as you should know, has 1 carbon molecule in it. Okay, so we have CO2 coming in, and this carbon is, of course, the carbon that's going to be incorporated into the sugar later on. So the RUBP binds to carbon dioxide, and for this step to happen, we need an enzyme, and we have a very significant enzyme here. In fact, it's the most common protein in the world, this particular enzyme, which is quite a feat. It's called Rubisco. And the reason it's so common is actually because it's not a very good enzyme. What do I mean by that? Well, it's not very good because it's slow. So plants have to make tons and tons of Rubisco in order to catalyze this reaction fast enough for them to survive and produce enough sugar. As a result, Rubisco is the most common protein, as I mentioned. What's interesting about Rubisco is it does something that we as humans aren't able to do, that is trapping carbon dioxide effectively, because it binds it into this molecule. Carbon dioxide is pretty unreactive as a molecule, yet plants are able to get it to react using this special enzyme. So the RUBP, which is 5 carbons, binds to a carbon dioxide molecule, forming a 6-carbon molecule, which breaks down into 2 3-carbon molecules, and these have an abbreviated name of 3PG. Okay, I'll just write the full name out here. That stands for 3-phosphoglycerate. Oh, I'll just move that across so you can see it. There you go. So 3PG, and you can think of the 3, it stands for the fact that there are 3 carbons in this new molecule. So that 6 carbon molecule that came over from the RUBP is now being split down into 2 3 carbon molecules. Then what we have is a series of reactions. I'll just zoom out a little so you can see this. Where 3PG is converted, let's move that down, into another molecule, which is also a 3 carbon molecule, but please note that these are different molecules from one another, called G3P. It's get very confusing, okay? 3PG, 3-phosphoglycerate, and G3P. That stands for glyceraldehyde phosphate. And don't worry, I'll just move this across so you can see the whole of that word. There you go. Glyceraldehyde 3-phosphate. Again, this 3 can help you to remember that there are 3 carbons in the molecule. That's not actually what it stands for, but you can think of it that way and that might help you. So this is G3P. And again, the long name, you don't really need it, but if you're familiar with it, that helps. If you know G3P, then you should be fine. At this point, some of the G3P is going to move in one direction and some is going to move into another direction. G3P can be converted into numerous other molecules. The one that you will come across obviously the most is, of course, our 6 carbon glucose. Now, as in all of these, we know that the 6 carbons in glucose aren't in a straight line. I'm just doing that way so that we can keep track of how many carbons are present. Now, if you're good at math, you'll realize that to make a 6 carbon molecule, we're going to need 2 of these molecules, 2 G3P. So I'm just going to write 2 times there to show that 2 of them are going in that direction. However, it is important to maintain this reaction that the G3P gets converted back into RUBP. To do this, we have a series of reactions, which I'm just going to draw with a dotted line arrow, like so. And you don't really need to know much detail about these at all. I'm just going to write a series of enzyme catalyzed reactions. Okay, a series of enzyme catalyzed reactions. There you go. So what is enabling this to happen? Because this doesn't just happen on its own, we need to put in energy. In particular, we need to put energy in these 2 stages here. To convert 3-phosphoglycerate into glyceraldehyde 3-phosphate, we need to use ATP in order to phosphorylate the molecule, and this produces ADP here. So I'm just going to write on the inside of the circle at this point, I'm going to write the name of this process. This is phosphorylation. Or phosphorylated. Okay, it is phosphorylated. Okay, as well as that, we need the high energy electron carrier NADPH. Remember, if there's a P present, we're talking about photosynthesis normally. And down at the base here, that is converted into NADP+. Because NADPH is providing some electrons to this, we say that it is reducing the molecule, and it's providing it with these high energy electrons as well as hydrogen ions. Now, it's important that you understand where these come from and where they're going back to. Both the ATP and NADPH have come from the light-dependent reactions. Light-dependent reactions. Okay, and both will go back there in order to be regenerated. So really the light-dependent reactions are driving this cycle. This cycle is known as the light-independent reactions, or the dark reactions, or the Calvin cycle, because it's not directly reliant on the energy from sunlight. But that energy is coming from sunlight just via the steps of the light-dependent reactions. So you can see here ATP being converted to ADP and NADPH to NADP+. We also need to use some more ATP in order to regenerate the RuBP. So again, these series of reactions here require ATP to be used up to phosphorylate these molecules, make them reactive, and those are converted back into ADP. Now, one thing you need to be aware of as you're looking at this is you need to be able to track the number of carbons that are going along. This is a higher level activity, but something you should be able to do. Let's think about this. In order to produce one molecule of glucose, we're going to need to have 6 carbons. Okay, that makes sense. Which means, if we think about where carbon is coming into this cycle, that we're going to need 6 molecules of carbon dioxide. Now you should remember that from your equation that you did to learn about photosynthesis. Now let's think about this logically. If we have 6 molecules of carbon dioxide, we're also going to need 6 molecules of RuBP. So I'm going to put a 6 in front of this so that I know that I've got 6 of these. If you're particularly good at math, you may be able to work out then that if I have 6 of these, 6 times 5 carbon, well, that's going to be 30 carbons total. These join together to make 3PG, and because it splits into a 3 carbon molecule, there's going to be 12 of them. Okay, that should make sense to you so far. This results in 12 G3P. Now I've already said that 2 of them are going to form glucose. The other 10 are going to be transformed back into RuBP. And let's think about this. 10 times 1, 2, 3 is 30, which not coincidentally is the same as 6 times 5. So we can know that we can regenerate 6 new molecules of RuBP, and so the cycle can continue. It is challenging to keep track of the number of carbon molecules, but if you've got that sorted, and if you understand all of this process, the fact it's happening in the stroma, what it requires from the light-dependent reactions, and what it is returning to the light-dependent reactions, what it is producing, ADP, NADP plus, and glucose, then you're all sorted. 6 carbon dioxides converted into 1 glucose molecule. If you can combine this with what is happening in the light-dependent reactions in the other video, then you should be able to come up with the entire formula for photosynthesis, which you'll remember is 6 CO2 plus 6 H2O is transformed using energy from the sun into 6 oxygen molecules and 1 glucose molecule, which is C6H12O6. Excellent.